Today I will be talking about one of the most fundamental parts of anatomy and physiology, action potentials. In a nutshell, an action potential is a change in membrane potential sensing neighboring neurons, allowing the transmission of electrical signals to move your body and send signals to and from your brain. But what does that even mean? Where do they come from? I'm so glad you asked. There are 4 key steps that need to take place. But first, we have to think about the electricity in our bodies. In a resting axon, the electrical charge is usually negative 70 millivolts. This is called the resting state, or polarized. No ions are moving during this state. The electrical gradient, alongside the concentration gradient, drives the ions to move either in or out of the cell during an action potential. Once our body either gets a signal from the outside world or signals from other cells in our body, depolarization occurs. This is caused by sodium channels opening, allowing sodium to flow into the cell, making it less negative inside the cell, or more positive. This ranges from negative 50 to negative 55 millivolts. With the influx of sodium, the membrane potential will become positive, plus 30, but only for a few milliseconds. This threshold during depolarization needs to take place in order for the action potential to occur at all. If the threshold is not reached, the action potential will not be propagated along the axon. Once the threshold is reached, the action potential either occurs fully or not at all. The amplitude of the action potential is always the same, regardless of the strength of stimulus. Next, repolarization will occur. This is caused by sodium channels closing and potassium channels opening, allowing potassium to leave the cell. This causes the membrane potential to drop down to negative again. The final step that occurs is called hyperpolarization. This is caused by potassium continuing to leave the cell, making the membrane potential even more negative, going back down to negative 70 to negative 90. I have written out how this is in a graph. So here you can see resting at negative 70, the ion channels open, the sodium channels open, causing it to go to positive 30 during depolarization, and then during repolarization, the potassium is flowing out of the cell, causing it to go back down, negative, and then hyperpolarization occurs because potassium is still leaving the cell. And then finally, it'll eventually return to the negative 70 resting state. And here's the threshold that needs to be reached for the action potential to occur. And some things to keep in mind about action potentials is that they differ from graded potentials in their magnitude, propagation, and function. Magnitude is always the same regardless of the stimulus. Propagation over long distances without weakening via Salvador conduction. And action potentials are an all or nothing occurrence requiring a threshold. They function in long distance signaling in nerve cells and in muscle cells. In summary, action potentials are very important for movement and sending signals to and from your brain. Without action potentials, nothing would get done. Speaking of muscles, action potentials are a key player in the initiation of muscle contraction. Here's Taylor to explain. Thank you.
Hi everyone, today I'm going to walk you through how muscles contract and the essential role sarcomeres play in this fascinating process. First, let's start by understanding that muscle contraction is a process that allows us to move, breathe, and even maintain posture. This process begins when the nervous system sends a message to a muscle. This happens through a specialized nerve cells called motor neurons. These neurons release a chemical messenger called acetylcholine at the place where the nerve meets the muscle, known as the neuromuscular junction. The acetylcholine binds to the receptors on the muscle surface, causing an electrical impulse called an action potential to travel along the muscle cell membrane, known as the sarcolemma. But this signal needs to go deeper into the cell to trigger contraction, and that's where T-tubules come in. These structures help the signal dive deep inside the muscle fiber. Now, inside the muscle fiber is an important organelle called the sarcoplasmic reticulum. Think of this as the storage center for calcium ions. When the action potential reaches the sarcoplasmic reticulum, it causes calcium ions to be released into the cytoplasm of the muscle cell, and that is where the magic begins. These calcium ions bind to a protein complex called troponin, which is attached to another protein called tryptomyosin. Tryptomyosin is like a gatekeeper that covers the active sites on active filaments, preventing myosin, the thick filament, from grabbing onto actin. When calcium binds to troponin, it causes tryptomyosin to shift out of the way, exposing the binding sites on actin. This is when myosin heads come into action. Myosin heads bind to the exposed site on actin, forming what we call cross bridges. Using energy from ATP, the energy currency of the cell, myosin heads pivot, pulling the action filaments inward towards the center of the sarcomere. This action shortens the sarcomere, and when many sarcomeres shorten together, the entire muscle contracts. This is known as the sliding filament theory, where the actin and myosin filaments slide past one another, but they themselves do not get shorter, they just overlap more. It's important to note that this process is highly organized. The sarcomere is the repeating structural unit of a myofibril, bordered by Z discs. When a muscle contracts, the distance between the Z discs shorten. Structures like H zone and I band get narrower, and the A band remains the same length because it represents the length of the thick filaments. Once the nervous system stimulation stops, calcium ions are pumped back into the sarcoplasmic reticulum using ATP. Without calcium, troponin and tryptomyosin return to their original positions, covering the binding sites on actin again. This stops the interaction between actin and myosin, and the muscle fiber relaxes. In summary, muscle contraction is a precise, highly coordinated sequence of events involving the nervous system, calcium ions, regulatory proteins, and the intricate structure of the sarcomere. Without this process, basic movements like walking, blinking, or even keeping your head up wouldn't be possible. So the next time you take a step or pick something up, remember, your body is using the amazing teamwork of the nervous system, calcium ions, and the sliding filament mechanisms happening inside millions of tiny sarcomeres. Thank you.
So, as I explained, the action potential is the electrical signal that begins in the nervous system. It travels down the neuron, reaches the neuromuscular junction, and triggers the release of acetylcholine. This chemical messenger is what allows the action potential to pass from the nerve to the muscle fiber.
And once that message reaches the muscle, that's where the contraction process I talked about kicks in. The action potential spreads along the sarcolemma and through the T-tubules, which triggers the release of calcium from the sarcoplasmic reticulum.
Without the action potential, the message would never get to the muscle, and calcium wouldn't be released.
And without calcium, the actin and myosin filaments in the sarcomere couldn't interact to cause muscle contraction. So really, the action potential and the muscle contraction are two parts of the same chain reaction, and they rely on each other to make movement possible.
In short, the nervous system sends the signal, and the muscular system does the work. Together, they power every move we make. The end!